back to India and did not come to any of the society meetings. So I thank Professor Pandey, Professor Nampi and many others who have encouraged me to come to the society meetings. But I hope I will justify why I was missing from conferences by the body of work that I'm going to present today from my talk and the 6 other talks that my students are going to present in this conference. So I will use the excuse that we were busy working at the Tropical Ecology and Evolution Lab to establish certain models which we can investigate for plant evolution and to understand diversity of plants in India. And it took me a long time to come to IAAT, but I'm honored to be here. I thank the organizing team, the organizers, the IAAT executive body who have supported me, Manu specifically for making excellent arrangements for me and all of us. I think we will all agree that this has been a fantastic meeting so far. And with this, I'll just take you through this a short journey of what we have discovered in the last 12 years of plant diversity and what data from some of these students, past and present, who have contributed to this project. So Ajit, my past students, Alina is here in the audience and will be giving a talk later today. And Anupama is, and so is Saket, who's passed out. And Anupama is here as well, will be giving a talk later today. So most of the work here is going to be highlighted on this particular image that you see in the center, which is the Hidicium. Hidicium is the genus which is the most, one of the interesting genera within Zingiberraceae, which has fragrant flowers. Of course, it looks very pretty, but what attracted me to Hidicium was that it had interesting floral traits. So when I came flora, which will connect ecology, evolution to systematics and floral traits and so on and so forth. So my email address and access is here for students who would like to reach out to me. Like Professor Pandey said, my lab is called Tropical Ecology and Evolution Lab, where we look at both ecology in terms of behavior in the wild and evolution that is plant systematics. So to begin with, one of the questions that we all have to address, because we've been discussing, almost debating quite strongly about what is a taxa. Now remember, when you're describing something as a biodiverse region, the fundamental element that hyperdiverse or biodiverse region, when that number is small, you say that nomenclature discussions that we've been having and so on and so forth. So the key question here is, what are the factors that drive this biodiversity and
or we should identify features that makes a specific region hyperdiverse, right? And that has been sort of the key motivation for most of the projects in my lab. So the question is, in this kind of a, what this essentially means is that we're already accepting that traits are variable, right? That is, when you have a species, traits are going to show some kind of differences or variations and forms. And if that is the case, then as taxonomists, you again end up at this juncture where you're going to ask, if traits are variable, then what is a taxon, right? What is a species? And that again, we get back into that argument of what a species is. Now, there are two definitions that we all know about, or multiple definitions of species. The one main definition of species that you know is from Ernst Mayr, which is the biological species concept, which suggests that species is a group of actually or potentially interbreeding natural populations which are reproductively isolated from other such groups. So this automatically means that there should be some kind of reproductive isolation for a species to be in its true form, and that's the biological species concept. This requires that there's some kind of reproductive incompatibility between what you call as two different species, and therefore there should also be lesser gene flow. The contrast to this, now this is kind of, if this is happening, it's a very stable system, but what we often see is what is called a species complex. This is a group of closely related organisms which are very similar in appearance, that so similar that the boundaries between them are often unclear, and therefore we heard that yesterday as well. One person says, I think this is a species, and the other person says this is not a species, this is native, non-native, and all these debates come on a taxonomic sort of background. What this species complex means is that there's reproductive compatibility between the individuals that you're examining, and also there's some kind of a gene flow. Now, if you look at what the species continuum is, and I think this is something that is very important for all of us to understand, while we give a name to a species, and we are assuming some kind of a stability and staticness, we all also should, I'm hoping we all accept as well, that species is a continuum concept. What it means is that if you take a phylogenetic tree of this kind, and you take a node, I'm not sure the pointer is working. Anyway, if you take a node like this, at that node, if you zoom in and look at how the species are operating, you can find the species in these three different forms. Hypothetically, you could be in the forest, you're collecting the plant, and the plant that you're collecting could be in this format, this one, or the last one. If you are in this format and this format, that is it, that species, part of that species continuum, you're possibly lucky. The best state is when you find the species in this differentiated form, that is, you have species 1 and species 2, and morphology is telling you that all is good and you can identify them as separate entities. If you find this is the polymorphism that we often see within a population, massive amount of variations in morphology when you're in the wild, and that's the initiation of what could be the species continuum, and this is a population level model, and the question then has to be addressed at a population level, not at a species level. What unfortunately in the genus Hidicium we found ourselves was in this most difficult space, that is in the central space, where differentiated populations are connected by some kind of a gene flow, which means that we are able to identify populations, and yet at the same time we are not able to draw a specific line saying this is population 1, this is population 2, this is species 1, species 2, and so on and so forth. So as taxonomists, as evolutionary biologists, we have to enter the forest with the idea that you could find your species either in this early phase of the species continuum, the middle phase, or the later phase. And if you know where you are, you can tackle the question of biodiversity or diversity better, because you can design your questions based on that assumption. So what I'm going to talk about today is Hidicium in this larger global region, what we call as the Indo-Malayan realm, and I'm going to refer it to IMR, and most of our publications refer to these regions as IMR. So Hidiciums are found across this entire region, and we tackle the phylogeny of Hidicium considering its vastness and distribution across the region. A quick recall, there's a Himalayan orogeny, that is how Indian northeastern plants are affected by how the Himalayan uplift operated. You know that the tectonic plate basically is in a subduction zone. It came, hit the Eurasian plate. India was a floating, let's say, arc, hit the Eurasian plate, resulted in what we see now as this massive, majestic sort of Himalayas, which are forming a barrier in some form to taxa that could actually come from China and Southeast Asia from the north. We also have this Southeast Asian sea level changes to deal with when you're looking at the entire Indo-Malayan region, where as recent as about 25 million years ago, the landscape looked very different. Like you can see here, there are a lot of interconnecting spaces. What we see now are these hundreds of islands across Southeast Asia, which makes it into a very, again, interesting but also evolutionarily a difficult question to solve, because the histories are not very, geological history is not very clear. So we kind of asked, when I started the Hiddekiam project, the main question we asked is, what is the role of the mighty Himalayan mountains in generating this biodiversity in the northeast? Because we've heard this yesterday again, that we're talking about Western Ghats as a biodiverse region, and then it's the northeast and the Himalayan region as the biodiverse region. So to quickly give an introduction, this is some of the diversity in Hiddekiams. I've arranged them by color, so you can see, we can see white colored flowers, a little pale yellow colored flowers, dark yellow flowers, and red colored flowers. So color is you can find all kind of colors in Hiddekiam, white being significantly dominant. When we started, we divided the entire biogeographic region where we had collected the Hiddekiams and were using in our phylogeny into the 7 major biogeographic groups, and this is what we have extensively now geologically studied and identified as regions that affect the biodiversity or speciation processes within Hiddekiam. So long story short, we took as many species as possible. So almost we did 90% sampling within the genus. The only few taxa that we are missing are key taxa from China and Vietnam. And we built this massive phylogeny here. This entire phylogeny from the color part are Hiddekiam. There are about 110 species in this genus. We used as many as possible, including population forms. And we built the phylogeny based on molecules. Of course, when we collected the samples, it was based on morphology because that was our identifier. And the long story short is that we found that the genus Hiddekiam is very young, and also a very sexy group. When I say in terms of sexy group, in terms of reproductive compatibilities and mating strategies that it has, which is quite open within the genus. So most of our papers have already been published. This is the first work where we actually showed that the Himalayan orogeny and the following monsoon sort of intensification explains the species diversification that you see in the group. And what you want to focus here is, this of course is the time series, so the x-axis here is time, and you can see here that something happens around here where the Hiddekiam just starts exploding, and the outgroup here are the rest of the gingers that we had used. The upper graph is where I'm going to go next, which is being used to explain why I say that Hiddekiam has been affected by the Himalayan orogeny. For the morphologists here, of course we looked at all the floral characters and vegetative characters, and if you're interested, you can go character by character. I'm not going to hang on to it, but I'm putting these slides just to say that while we are molecular biologists, we use molecules, we also emphasize on morphology because it's really not possible to not use morphology. So the argument is really redundant for me to that whether one should use morphology or not, you will end up using if you're describing a plant and you're interested in the plant, because we're not identifying or recognizing a species because of the ATCGs, we're recognizing it because of the morphology that we are looking at it. So we did all of these analysis, and we found that morphologically there was no synapomorphy that we could find for any character across the phylogeny of Hiddekiam. We also did a lot of correlation analysis to see if characters correlated and could identify features within the tree. And all you have to see from far is that whether the left side tree, the dots, the color matches or the pattern matches with the right side tree in both of these panels. And you can see that there is some kind of a matching that you see. For instance, you can see there are yellow dots here and yellow dots here, and then in the upper part of the clade also seems to match. Now the traits that we had mapped here is slender cylindrical inflorescence and coiling of the floral tube. Why did we look at these morphologies? Because we had an inclination and an idea that these traits may be correlated, and the phylogeny, or mapping it on the phylogeny, was our way of saying that there is possible features that are correlated, resulting in sort of clades and the structure of the clades based on morphology, if one wanted to look at it. But the trees were built on molecular data, so we just basically mapped the morphology to look at correlations. So the big question for us was this phylogenetic structure, and what we discovered were there were at least 4 major clades. We have now revised it in our recent paper that just came out yesterday, and what the take-home message is that there are 4 clades, and one of them here, from the size of the triangle, you can say is the largest. We're calling this the Coronarium clade. Coronarium is also the type species for the genus. And it's the most speciose. The second is the villosum clade, slightly smaller. And then there's clade 1, 2, the spicatum, the densiflorum, and the ellipticum clade. Overall, the number of species in this clades gets smaller and smaller. So there is something going on here in clade 4, which resulted in this massive increase in number of species that increased in the Coronarium clade. When we mapped the geology of the Himalayas, like I said, we knew what the subduction zone is. We know the rate at which the Himalaya uplift is happening even now. We found that when you track here, the x-axis is the time. This is the millions of years. And the y-axis is the number of taxa. So you kind of add species year by year in millions of years, and you see if the accumulation shows a trend. And what we see here is that something happens. There's not much of species accumulation till 4 million years, and then you start seeing that the species, the number of species in this genus has started increasing slowly and steadily, and it continues to increase till almost like a plateau that it has reached now. When we mapped the geological and climatic traits, 2 trends came out, or rather 3 trends came out. The Himalayan uplift is supposed to have occurred a little before the 4 million years. What that uplift means is that it created possibly ecological niches because more geographic spaces were created with diverse traits in climate. The uplift also resulted in an intensification in monsoon, and we know that most gingers like water, they like humid environment, and therefore the environment changed, and these 2 kind of resulted in the slight increase in the speciation rates that we see within the genus. There was a mild glaciation period also later on, and so therefore there were clearly climatic shifts that were happening across in the last 8 million years, which we now think has resulted in this species diversification within the genus. So once we had that, we of course went on to see the biogeographic patterns in it, and what we found is that the center of diversity for it is the Indo-Myanmar region. So somewhere in the center is where the Hiddekium, the base of the early diverging group is, and from there it moved out into India, also into Southeast Asia. The triangle, the size of the triangle tells you how many species have been contributed from this specific region. So you can see that the largest diversity is in the northeast of India, possibly fed in from this Indo-Myanmar region, and the second largest, is into Southeast Asia and so on and so forth. And there is one trajectory that also came all the way to Western Ghats. This arrow should not be over the water. I'm suggesting that it came through water. It came somewhere through land, so I could not change the arrow, but consider that the plant here came somewhere from the land. And Alina is going to talk about it using her genomic data, so you can hear more about it later. So the first conclusion is that the Himalayan orogeny shaped the diversity of this understory ginger, Hedychium, and once it arrived on the Indian subcontinent, and this pattern has now also been, the pattern I mean is the biogeographic pattern, and the dispersal pattern is now been seen in Globa, in Didymocarpus and few other species as well that we're working on in the lab. So the next question becomes, so what happened to Hedychium once they arrived to the subcontinent? Because remember I said that among the 4 clades, this first panel shows you clade 1, 2 and 3. The second one is the clade 4, which we call as the Indian clade or the Coronarium clade. And if you just purely look at the number of terminals that you see, you can see clade 4 has more terminals, that is more number of species. Clade 1 to 3 has combined less number of species. So our immediate next question was, what is happening with this clade 4? Again, going back to the first question I had asked, what is biodiversity, or why is this clade more taxonomically diverse for that matter? So we kind of went back and tried to think about how is this diversity created, why is clade 4 having more taxa, and is polymorphism, the idea that a species individual can be in different morphological forms, got anything to do with the diversity that we were seeing? And this led us to investigations into reproductive trait diversity, mutation rates, and so on and so forth. So just to give a quick idea of what polymorphism is, this is a different project that we kind of almost concluded it, but it's still going on from the molecular end of it. This is the Curcuma caulina, which is from the laterite plateaus. And on a single plateau, you can see all of these variable forms. So when you talk about biodiversity, polymorphism plays a very important role because we need to know where to draw these lines, or can we draw the lines when we look at these polymorphic traits within a species. So, of course, if anyone is as old as I am, you probably remember this song. So we went back, Ajit and I, we looked at clade 4 and said, why is clade 4 so diverse? How can we actually explain this diversity? Is there anything we can do to explain it? And the answer to this is very simple. The first answer is that promiscuity is good for diversity, and of course I'm putting asterisks in plants because this is not an explanation for animals, and if there are students here, this is not propagating about promiscuity, but yes, when it comes to biodiversity, it helps for plants. And sympatry helps promiscuity. That is, if you are next to each other, it helps in this reproductive sort of lability that organisms show. So, and this is just again to give you an example of what is happening to Hedychiums now within northeast of India. So in Arunachal and in Nagaland, if you go to several locations, you will see that the upper part shows what you see in the wild. On the left of this image, you see Gardnerianum. On the right, you see a species called Coccinum, and in less than 100 meters, you will see these intermediate forms. Don't take pictures and don't go and describe these species. That's not the point of this image. In fact, if anything, the point of the slide is do not describe these individuals, these intermediates, as new species, because we think there are intermediates and there are hybrids. And unless you will get it published somewhere, I have no doubts about it, but that will result in taxonomic problems and chaos for some of us who are trying to understand how this genus has evolved. Similar situations exist in Nagaland between Marginatum and Stenopetalum, and we are calling it a complex because these are intermediates. In less than 50 meters, you will see Marginatum, Stenopetalum, and intermediate forms, and you can see the morphological structures look different enough that a morphologist may call it as a different species. So we looked at these morphological variations of intermediates because we wanted to identify, are these different species, can we call it different species, without actually using molecular data. And so we published this using some kind of a machine learning model because the intermediate forms are very, very complex and the overlaps in the morphology is quite high. So we ended up, we realized that we were sort of somewhere in this middle zone where we kind of morphologically can tell these are different looking species, but we were not able to identify which trait I would use to identify it. So I had a student, Preeti, who was interested in using computational methods to form clusters, and we ended up publishing this work. And we found that the hedychiums in the northeast are in this intermediate step towards speciation. So if you go to the northeast, you will find them in this. So therefore, it looks like hedychium is hyperdiverse, but it's primarily because it's quite a young group and it's intermixing. Do I have time? Yeah. Okay. Oh, conclude, okay, give me 2 minutes. So this is the work from Preeti, like I said, where we looked at the species complexes, and because we wanted to understand how the speciation is happening in clade 4, we took then this only one species from this entire group, spicatum, which also forms massive amount of intermediates in the northeast, and we created a model out of it and have been studying it for the last almost 6 years now extensively to understand what is going on. And for this we looked at sympatry first, like I said. I already told you that sympatry causes species complex. So what we did is we've now built this massive networks. So I'm just highlighting here spicatum. What this connection means is that whenever we've found spicatum to be sympatric with all of the species with which it is connected. And these are field data sets that we are getting. We looked at phenology. Not only is it sympatric with these species, it's also flowering simultaneously with all of these species that is shown in the network. And finally, it's together, it's flowering together, and is also reproductively compatible. That is, if you did a cross, you will get viable seeds, and we did those crosses in both directions. This is the maternal and paternal, both directions have been done, and we now know the reproductive sort of networks of what spicatum is compatible with. And so the long story short for this is that living close by, flowering overlaps, and absence of compatibility barriers results in what we call is this genetic diversity as well as morphological diversity in these hedychium groups in the northeast of India, justifying what I said before, that promiscuity is really good for diversity, and sympatry helps this promiscuity because you're close by, chances are the pollinators are making mistakes and therefore resulting in reproductive sort of fertile outcomes. So there are a couple of talks from my lab for more stories on this project. There's Anupama talking on floral volatiles because gingers, the hedychiums are very fragrant, so we looked at also the chemotaxonomy of it. Abhishek also looked at essential oils, and Alina, who's going to give you the long story for the genomic story. This is Alina's work where we then looked at spicatum and used genomic data to see the population level variation, and you can see here within the northeast, each population has its own genotypic signature and is quite different from each other, which is now we can now collaborate with or corroborate with the ecological work. Right. So the conclusion, the second main conclusion from our study is that when morphological diversity makes taxonomic identifications complex, it could be a result of at least 3 non-exclusive factors, that is phylogenetic age, which is the gingers are a very young group, or hedychium is a very young group, dispersal history and sympatry, and lack of these reproductive barriers. And this is just the tip of the iceberg. Hedychium is super complex in the northeast because it's not a linear speciation, it's actually a triangle, it's a hexagon, and it's very, very complex now. And this is basically the hybrid swarms that we see all over in northeast. So taxonomically, it's a very difficult group to work with, almost as bad as impatiens for that matter when it comes to species. So with this, I'll sort of end because I don't want to take more of the time, but we've done similar studies now with curcuma, where that polymorphism has also been shown to be because of ecological reasons, and reproductive barriers are present even within the same polymorphic species, different morphs within the same species. And there are another 3 sets of talks from my students who have worked, Rutu, who's worked on the enantial style in Didymocarpus, and also the stories from laterite plateaus and the Himalayas, where we have looked at plant-pollinator interactions to explain why speciation is happening in the way it is. So to conclude, the definition, I started off with this box from the New Phytologist paper, and what we find in Hidicium is that we have this few taxa which show high gene flow, few taxa that show low gene flow, and the low gene flow ones of course have very distinct species boundaries. This taxa which show high gene flow have indistinct species boundaries, that is you have this species complexes. And the one feature that is that axis that explains it is the distribution and sympatry. The second axis is phylogenetic age, and the third axis here is reproductive compatibility, resulting in some of these taxa showing hypervariability, hypermorphological variations, polymorphism, you name it, and intermediates and hybrids. With this, I'll thank many collaborators that I've been lucky to have. Some of them are here. With Michael, I've been working on Didymocarpus phylogeny, and a lot of people from Southeast Asia who have contributed samples, and all the forestries, and of course my brilliant students, and I welcome anyone who would be interested to collaborate and work with us at ISER Bhopal. So thank you.
Thank you, Dr. Vinita Goda, for wonderful presentation. And starting with the term taxon and how we should define, and especially now we know much more about how the speciation takes place and how angiosperms are so diverse, especially taking into account the example of Hidicium in Indian Himalayan region. You have shown that how the Hidicium different species have evolved, and there are many intermediates, and those who are visiting Northeast region, they should not be in hurry to publish a new species. So I think here is a caution, because many of the taxonomists, we personally feel that we should publish more and more new species. Also it is important because many species may vanish forever if we do not describe them. So, but definitely we should not be in hurry, and a lesson is that you should take into account other data also. Besides morphological characters, there are many other characters. She has taken ecological characters and molecular data and other aspects, and she has shown that how diversification has taken place, and especially the flora evolution and diversification in this, and also she has given the example of the Curcuma caulina. And Vinita, you have coined the term sexy ginger.
Yeah.
So can you explain? Normally sexy word we do not use in plants.
Yeah.
But that's why. Okay. So
yeah.
We would like to know, the audience would like to know that you have coined a term and which is very attractive.
Yeah, so our second paper, the AJP paper, we start the paper with a quote from Darwin. So it turns out that Darwin received a sample of the Hidicium as a herbarium sample, and he looked at the flower and said, well, I know there has to be a moth, similar story to what he said for the orchids and the orchid moth, that there has to be a moth which is, pollinating these Hidiciums, and that the moth is what is driving this long floral tubes of this genus. And then he realized that it was fragrant, so I said, of course now it has to be a moth, because it's not only long and tubular, it is fragrant, it's nocturnal, and so on and so forth. So the speciation of Hidicium was always expected because of the floral reproductive traits, hence the sexy part. So we thought that, and I do think from our data also we see that all the taxonomic issues we have is because of reproductive compatibilities. So the reproductive sexiness is super important for the species, and what's happening now, or what we have seen, interestingly and disappointingly, the only thing we have not found in Hidiciums in the Northeast is the pollinator. That's the only thing that we haven't found. Why? I have no clue. But what is happening is that I think the principal pollinator is missing. It has been possibly replaced by secondary pollinators like the bees, and the bees are not very selective when they go and forage, resulting in this intermediates and high hybridization that we see, because they're not specific about who they're pollinating. So the absence of a true pollinator possibly is causing this taxonomic problem, and therefore all of it is about reproductive compatibilities in ginger. So that's why the sexy ginger. Yeah.
I think we all should take into account the reproductive biology data in systematics, because here lies some important questions which are to be addressed.
Yeah.
So Dr. Pandurangan, you have something to say?
Yeah.
You have shown that Himalayan realm,
Yeah.
but that is boundary extended to by merging with Malayan realm up to
Correct.
Asia Pacific.
Correct.
The plants can move
Yes.
from one realm to another because
Correct.
the boundary of the realm is later on only we have actually
Yeah.
more demarcated. But why you extended up to Asia Pacific by mentioning the Himalayan realm?
Because I think
That's third or fourth slide.
yeah, we can go back to it.
No,
if
no, no,
someone- yeah.
please.
Yeah, so I can talk to you in person about it now that I know the question. But yeah, the realm and even the biogeographic regions we have redefined for hedychiums. Let me be clear about it. And we took a lot of time and effort to redefine these biogeographic realms. Because most of the Asian regions, the geology has been studied by the global sort of north, colonial sort of people, and they have defined boundaries that actually is not substantiated anymore with new geological data. So it is important for us to redefine these biogeographic regions. So if you see our biogeographic regions for hedychium is also, there are 7 regions, not 4. So there's a lot of things that we redid it because I think we need to rethink these things. Yeah.
Yeah. Do you know that Idikium is referred even Mahabharata actually?
Yeah, I'm sure.
Dr. Vinita Goda will be here, so we can take 1 or 2 questions. Okay, quickly, please. Yeah, I think she will be here and
Yeah, thank you very much. Actually, I just wanted to intrude. It was really excellent, and you showed us this, the new species is not the way, and getting into the whole topic of understanding it is a wonderful thing. And that's why in the morpho taxonomy, what we do actually, it is that actually whenever you identify a species which doesn't actually fit into a species, and it is an intermediate between the species, where we actually go for the eco-species types, varieties, and all those things. See, as much as it is very important that we should not describe as a new species, but identifying those species as slightly different from the other species and which populations are there is also very important to have a record of those things. That's why many times what happens is like Rhizancia, Ceciliflora variety, this thing, or subspecies, this thing. So like that, in morpho taxonomy, we keep those species. Earlier it was a lot of things like that.
Okay,
Nowadays,
Dr.
after the genetic this thing is as compared.
I think you can discuss with her during the tea time.
Yes, yes.
The last question.
Thank you very much for a very enlightening lecture. It shows your in-depth work and understanding of the subject. Madam, I just wanted to add one thing. This is not a question actually. Hedychium is a very popular Ayurvedic medicine. One preparation called Chardiripu is produced out of the tumors of this. It is anti-emetic, very useful for the pregnant ladies, those who are having the morning sickness. And Cochin and Kerala had been said that they were seafarers, and that shows its Dutch connection and the other one also. So probably that has come as a medicine over here and spread. Maybe it may not be directly from Himalaya. This is one of the routes. This is just our suggestion.
Possible, absolutely. That's a great point. It is
Because
very
even
much
the
possible.
Frankenese and other things we used to get from Malaysia, Jakarta,
Yes.
other things, the Loban, other Ayurvedic medicine.
That's a
Okay,
very good point.
thank you very much. Thank you, Dr. Vinita, for excellent presentation, wonderful work she has been doing and her associates, and let her congratulate her for her excellent presentation. Thank you.
Thank you, ma'am, for your wonderful talk. You are really an inspiration, especially to girls, to conquer the boundless world of angiosperm systematics. Keep shining and breaking the barriers.
May I invite Dr. A.K. Pandey and Dr. Satish George to felicitate Dr. Vinita Gowda. Thank you all. We have important announcements. Those who are yet to submit their presentations are requested to contact the audiovisual desk at the earliest. I repeat, those who are yet to submit their presentations are requested to contact the audiovisual desk at the earliest. Participants who have opted for the conference t-shirts are requested to reach registration desk in front of the APJ auditorium today to collect their t-shirts. And those who wish to purchase the conference t-shirts but did not opt for one during registration may also register now. Distribution for these additional requests will be done on a first-come, first-served basis, subject to availability. Thank you.
I have great pleasure in introducing Dr. Santosh Nampi, who needs no introduction. Everybody sitting in the audience, everyone knows Professor Nampi is an outstanding plant systematist who has been working in the area of floristics and also pollination biology and the cytology and all other aspects. And of course, he has published over 110 research papers and 7 books and guided 16 PhD students, and he is fellow of the Linnean Society of London, and also he is recipient of V.V. Shivarajan Gold Medal, Birbal Sahani Birth Centenary Medal, and so on. And one thing I would like to add, that this year he has been elected as the fellow of the fellow of the National Academy of Sciences. So congratulations for that. And now I will not go into the details of his biodata, so I just invite Professor Nampi to deliver his talk.
Okay, now it's okay. So, good morning everyone. I am delighted to speak on the evolving landscape of plant taxonomy in India. And I specifically speak on integrative plant taxonomy. Yesterday we have a detailed discussion on omega as well as alpha taxonomy. There are a lot of discussions. But we know taxonomy is only one. The purpose of taxonomy is to provide a stable classification. And what all classical or molecular taxonomy actually means they are only evidences supporting to provide a stable system of classification. So in my talk, I may focus on how to integrate data in providing a more stable system of plant classification or plant identifications. Oh, reduced still. Okay, thank you. So we know India is mega diverse country with around one of the 10 top mega diverse country having 18,000 plant species recorded from India with considerable number of endemism. And we know despite enormous work done by Indian taxonomists, especially in the level of documentation, exploration, the understanding of many of these plant groups are not clear, or most of the plant groups are inadequately understood. So here is the focus of my presentation, to how to resolve these ambiguities in plant identification by providing more stable information on the identity of each individual species or genera or families. So there are a lot of issues in classical, while we are following the classical approach, there are a lot of issues, especially with respect to the morphological plasticity as well as cryptic species. So one of the place where the classical approach faces is with respect to the morphological plasticity or the cryptic, to document the cryptic diversity or of hybridization or of convergent evolution, etc. So in all these aspects, we know the inadequate type divisions is also one aspect, and again the limited field collections or scattered expertise are some of the area we fails to document the accurate identification of plant species. So what we actually mean by integrative taxonomy? Integrative taxonomy means it is not replacing morphology. We know the classical taxonomy is mostly based on morphological evidences. So here integrative means it is not replacing morphology, but it is enriching it with additional data sources. So what all we can integrate? So we can integrate all data sources. So we are integrating different independent lines of evidences from morphology, from molecular, cytology, anatomy, and other fields. Ultimately, the purpose is to provide the stable system of classification, and also we require plant species to be properly identified to avoid redundant descriptions. That's the purpose of integrative taxonomy. So I will speak a little bit about one classical example where we have integrated different approaches for resolving one ambiguity in the identification of ringoglossum. So we have revised this particular genus for Sri Lanka as well as India. This is a collaborative effort involving people from Sri Lanka as well as Professor Muller is also sitting here. So ultimately the purpose was to revise this particular genus for Sri Lanka as well as India, South India, and also to clarify the nomenclatural issues persisting in Ringoglossum scabrum. This genus was represented world over by 13 species, and India has 4 species, out of which 3 are occurring in South India. So here, the 3 species in South India are Ringoglossum notonianum, Ringoglossum obliquum, as well as Ringoglossum scabrum. This Ringoglossum scabrum was established in 1850 by Dalzell from the Konkan belt, and soon after its establishment it has been treated as a variety by Hooker, and later on it has been reduced as a synonym by Burt. But our integrative approach by combining multiple gene regions and also by extensive field survey, and ultimately we were able to re-establish the species status of Ringoglossum scabrum. So here we can see the distinctiveness of the morphology. See, this is Ringoglossum gardnerianum, while this represents Ringoglossum scabrum, and again we can see the peculiar nature of the inflated nature of the persistent calyx, and also the molecular distinctiveness also. So based on all these data sets, we have reinstated Ringoglossum scabrum as distinct. Another one, we are also working on another genus called Utricularia of the Lendibulariaceae. This is one of the group we are extensively studying for the past 6 or 7 years. 3 students are also working with me on different aspects of this genus. So here, we have revisited the section Nigricentus. This section Nigricentus is represented world over by 4 species, but in India we have 2 species, one of which has been treated as a synonym for long. So we have reinvestigated the particular species group with respect to the morphology, with respect to the micromorphology, and with respect to the molecular data sets. Ultimately, we reinstated that particular species as distinct based on the evidences. You can see the distinctiveness of, you can see the distinctiveness of the characters. You can again see the distinctiveness in the molecular data, and again on the micromorphology of seeds. Another one we have, another one we are working on the Commelinaceae for almost 30 years. I think we have started working on Commelinaceae from 1998 onwards, when I have a chance to work with Dr. Faden at Kew Herbarium. So that kindled my interest on working on Commelinaceae, and over the 30 years we are working on Commelinaceae. 3 PhDs have completed. And one student worked on the morphological aspect of Mordania, while other students have done the molecular phylogeny. And also during that time, we also worked out the cytology of several species. So while working on this is a new report for Murdania vaginata glabricapella. Initially, that species was described from Sri Lanka, but in the protologue, they written the description of that particular species is extending to India also. But there are no report of the occurrence by material from India. While our explorations in South India reveal few specimens, and while providing an accurate identification of that material, we done the extensive morphological analysis based on the field-based data, then again on the micromorphological data, as well as cytological, as well as the molecular data. We ultimately established the occurrence of Murdania vaginata glabricapella in South India. So that's one part of my presentation. Then what we need for empowering taxonomy in India. We need an integrative approach, not by working singly, but we need integrative people, integrative collaborative effort. Ultimately, be bringing more clarity in the taxonomic approach. So another one, empowering taxonomies. Integrative taxonomy also requires integrative people also. That means people who are expertise both in field as well as lab. So at present what we saw is those who are able to work in the lab are not a good field botanist. Those who are a field botanist are not expertise in the lab. So blending that is a must for safeguarding taxonomy in India. So there's another, so we need specialists for plant families, we need specialists for plant genera, and similarly, we need persons who can expertise both in lab as well as the field. So that's the need for the next. So these are my students working in the field. I am also sitting on the left in the field. So on the right you can see the students are working in either in the collaboration, in collaboration with other labs or in our lab also. So another one. So through collaboration, what we can achieve? Through collaboration we can have access to international overseas herbaria where we need to study the classical collections housed in various herbaria and also to study the type specimen physically. Otherwise, even though we are accessible to the digital specimens, what we require is the physical examination of specimens which are housed mostly in the overseas herbaria. So that can be achieved through international collaborations. Then revisiting the protologues ensures stability in plant names as well as identification. So we require correct identification of plant species as well as correct documentations and also for avoiding the redundant descriptions also. So we require identification, correct identification for stabilizing the plant nomenclature in India. So here we can see the collaborative projects accelerate discovery. So we are also collaborating at the initial time, we are also collaborating with the Institute of Botany Chinese Academy of Sciences for a visit there. And also many of our students also work with Professor Moller as part of the Gesnercy project. Then we have a collaboration on the Melastomyceaceae, particularly with Sonarla with Nicosalinis of the Florida Herbaria. So here we can, through this joint efforts or joint collaborative work, we have access to the materials located outside India, and also we are able to study the facilities available in their labs, and also we are getting more and more data sets for sharing, etc. So why does integrative taxonomy matter? Here we can see, so this integrative as well as collaborative approach will redefine the biodiversity estimates. And again, if we have a properly identified taxon, we can go for the proper conservation strategies. And it also, as Vinita said, it also goes for ecological as well as evolutionary research, aligned with national as well as global sustainable goals. So what is road ahead? We know we have a rich diversity in India. What we lack is we are working in isolation. So we have to go beyond that. We have to work in group, interact with, collaborate with others. So that means not an international collaboration, not alone, but we can collaborate with other national labs also. Now we have very good facilities in the national institutes. So we can collaborate both at the national level as well as the international level, bring taxonomy to be more refined for the betterment, for the sustenance of the biodiversity of India. So here we can bridge classical expertise with modern technology. That's a need. So yesterday also we speaks about alpha taxonomy at one end, omega taxonomy at the end. So both are required. We have to integrate. We know that, especially while speaking yesterday, Dr. Narasimhan while speaking his inaugural speech, he speaks that means how we can identify plant families based on APG. That's true. For identification of plant families require morphology. So is some of the earliest known classifications required. So, but we require modern technologies to resolve more data to identify, to strengthen the identification, especially for the identifications. So integration is essential. That can be achieved through either through collaborations or through either collaborations either through national or international labs, etc. So I will quickly go through some of the works we have undertaken in our lab. So we are working on the Gesneriaceae. For many years, 3 students are working on Gesneriaceae, Mr. Agil, Mr. Jenish, as well as Wahida. They are working on different aspects. Now recently we are studying the Jardonia indica. One of the Jardonia indica is one of the endemic species on which we are now presently working on the seedling morphology, the developmental aspects as well as the pollination mechanisms, etc. And also along with that we are also working on another endemic species. It's Hengelia prodebiana. So that student, Wahida, is working on the 2 species. Sorry. So as I said, we are working on the Utricularia for 7 or 8 years. So we have 4 students working on it, just Ms. Krishnapriya, then Vaishnavi, then Sneha, as well as Sreya are working on it. So we are working on this. So Krishna worked on the molecular as well as morphological aspects, while other 2 students, one student is working on the ecological aspect of the traps, while the third student is working, third and fourth students are working on the trap architecture through this. So this is supported by the ANRF, so on which we are working on the trap architecture. We are also working on the Melastomataceae for quite a few years. So Rashmi is working on that one, and we are also collaborating with Nikos Selnis in that one group. Then, as I said, this is a very interesting and passionate group for me. We are working on that almost 30 years on that group. So several students, only 2 students are presently working on it, while almost 3 or 4 students have done their PhD on Commelinaceae. So we did on the morphology, then cytology. As we are doing the cytology also, as well as some students have worked on the anatomical aspects. This is one group we have started recently. Arishma is working on that one, on the molecular phylogeny of Ixora. It's a very difficult group because there's 3 species, mostly of the 3 species. So this is the one group that we have recently undertaken. Cato is sitting here, Professor Cato. So we are working on the Podostimaceae. This is one interesting genus because it's a lot of interesting features can be seen on Podostimaceae. As we know, they have adapted to aquatic, they are the only aquatic angiosperm with wide distribution in South India, adapted to fast-flowing freshwater habitats and unique features. So a lot of unique features are seen in Podostimaceae. And 1 PhD student and 3 PG students are working on Podostimaceae, on different aspects of the Podostimaceae. And in a short period, I think Ashtadhar has made an extensive collection both in South India, and in the last week we explored some parts of the northeastern India together. So this is the overall our group at Calicut University, and thank all the audience for your patience and attention. Thank you everyone.
Thank you, Professor Nampi, for a very thought-provoking lecture that we should integrate different data sources in taxonomy to come to a particular conclusion. And in a true sense, integrative taxonomy is the demand of the day. And with many examples he has shown that we should take data from morphology as well as the seed morphological characters, our chromosome data, and pollen morphological characters. They are all important in taxonomy, and we should do the home message is that we should do integrative taxonomy. I have been working in India and also collaborated, but what we find that we are working in isolation, and there is a need for collaboration both in India and with foreign scientists, because many of the techniques and other things we do not know many a times. So it would be better that you collect the material here. Of course, we know National Biodiversity Authority has certain restrictions for sending the material, but of course, you can extract the DNA here and get the amplified product, or even sequences you can send, and it can be integrated at the with your collaborator outside country. So thank you, Dr. Nampi. We will not invite any question because whatever he has delivered, everyone knows, and we had a panel discussion also about this. So thank you very much.
Thank you, sir, for your wonderful presentation. Your insights and contributions to taxonomy are truly appreciated. May I invite Dr. A.K. Pandey and Dr. Satish George to felicitate Dr. Santosh Nambi.
That belongs to him.
And now, as Professor Santosh Nambi bids farewell to his teaching service in May 2026, his students have come together to honor his unwavering support, guidance and inspiration with a special token of love and appreciation. Sir, you have just retired from teaching service, but your legacy will continue to inspire us.
Yeah.
And now,
Yeah.
I would like to welcome the vibrant students of Professor Santosh Nambi to the stage to
Okay.
make this
Okay.
moment
For
truly
the-
special.
okay.
Please come forward.
So many times when we are going to retire, nobody wants that we should be known that retired person. But here is celebration that Professor Nambi is going to retire. Great.
I request Krishna Priya to reach the venue. I also request Tanya Francis to reach the venue. Awards to honor the best paper and post awards to honor the best paper and post presentations under the general themes. These newly introduced awards aimed to encourage high quality research, promote academic excellence and